we go. All right. This is Richard Nikoley, freetheanimal.com. And I have with me today Mr. Karl Seddon, over from out in the UK, London, I guess, right? Or Oxford?
Manchester, actually, at the moment.
Manchester. Okay.
Yeah.
Okay. So now the reason I'm having Karl on, I've worked with him for a number of years, I think 6 years now, since early 2015.
Yeah, 2015.
And, that's because he manufactures probiotics, and longtime readers of the blog Free the Animal know that I've delved into various aspects of gut health quite a lot, including probiotics and prebiotics. Karl manufactures a probiotic called Elixa. If you go to freetheanimal.com, you see it, and it comes to you in a box exactly that size. That's actual size.
And, yeah, that's right. Yes. You were actually blogging a lot about prebiotics in the form of resistant starch.
Yeah.
When you first came onto my radar, so it was around that time that I got in contact with you.
Yeah.
2015.
And so you've got your thing on the back of the box, so it's all professional, and then it says read me first, and you have a insert like that, and then you have 6 of these individual packets. That's a daily dose, and inside you will find 10 capsules. So there's 2 kinds of packaging, and there's science in both of them. There's packaging in this to keep moisture out, and then the actual 10 capsules are not just a gel cap. It's a very special thing that is made it's made so that when you swallow them, they don't just dissolve in your stomach and kill all the stuff, spores or whatever. I don't know. Karl can explain that. He's the expert. So
that's actually spot on. There are two layers of protection. The sachet itself has it's triple laminate, and it has a central layer, which is aluminum, which is fairly common to use to prevent the ingress of humidity and oxygen from the atmosphere.
Yeah, I guess people, things are even plastic, I guess, has some level of permeability in it. I don't know, but
yeah, it's surprising. It's very surprising the amount of atmospheric moisture that can get through a standard capsule and through a food container which doesn't have what's called a total protection to O2 and H2O ingress, such as aluminum.
Well, I guess that's why if you put something in plastic in the freezer, it's like, where did all that frost come from after it's been in there for a couple of months or something like that? I guess that's the story. So, Karl, what got you into gut health in the first place? I mean, or how did it get started for you?
So from a young age, probably around the age of 12 or 13.
So like 40 years ago?
I began having the typical like teenage skin. It wasn't too bad, to be honest, but I was having the typical like teenage skin, a bit oily, maybe some pimples here and there. And people would say, or my parents and adults would say, oh, it's a teenage thing. It's one of those things. And it always hit me as a very odd way for an animal to evolve, that there would ever be a period of time where you started to display what essentially looks like a contagious disease on your face. And so I never liked the explanation of, oh, it's just teenage acne or whatever. And that began my interest in looking at what exactly is it about our life that gives us these problems which animals don't face. So, for example, why do so many people have epilepsy and animals don't? At least animals in the wild. Why does almost everyone has some problem, whether it's short-sightedness or a skin problem or an allergy? Any animal in the wild that had myopia, like short-sightedness, they don't have glasses, so they would just be a goner. There's no way that a species in nature would evolve to have such a high incidence of problems such as short-sightedness, for example. They could potentially have, you know, the occasional 1 in 10,000 genetic defect, because that doesn't affect evolution too much. But evolution would absolutely annihilate, you know, the problem of teenage acne, whereby, you know, people at their most, you know, a period of important development psychologically would be presenting with something that appears to the other members of the species as basically a contagious disease. That's why acne looks, to our, you know, primitive brain, it looks unattractive. So I was very interested in what exactly is it. And as most people like yourself and other people, the first thing to look at is diet. What exactly is it about the diet? And then you go down the whole rabbit hole of insulin and insulin resistance and all these things. And I did a lot of self-experimentation, and there were, you know, you have inputs, you change the inputs and you have different outputs. But in the middle, the algorithm, the black box algorithm was clearly much more complicated than simply, you know, decrease insulin or something like that. So I was like, what is this black box algorithm? And at some point or other, I started investigating the idea that the actual black box inside that determines how your diet affects your health is the gut microbiota. And when I started researching that, I answered a lot of questions about just why it's so complex, why there's so much lag involved, depending on when you eat something, what happens as a result, and the whole myriad of different type of problems that can occur in a human. So, yeah. You
went on to get a science education, if I'm not mistaken, at university.
That's correct. Yeah, I did a degree in engineering science, but it was a master's degree as well. So in the master's portion of it, I did biomedical engineering and also process engineering.
There you go. Well, so you and then you went out and found a use for it, right? So now in probiotics, well, first of all, we already talked about your packaging and stuff like that, which is probably nobody else does this. I think there's like I've heard of like prescription type probiotics that are encapsulated in such a way as to get down to the colon.
Purportedly.
What?
Purported, yeah. I mean, okay, so there are very, very, I have never seen a probiotic which is packaged like mine, because it's capsules in a sachet. You might see powders in a sachet, but powders have such a high surface area and have no protection whatsoever that as they move into the stomach, they'll be basically enveloped in stomach acids and bile salts in the duodenum. So as far as the capsule is concerned, enteric coating is done in two ways, basically. You can coat the surface of the capsule, which you could think of it a little bit like spraying candy solution on top of an M&M. The way that they have the chocolate ball and then they spray the hard coating.
So it melts in your mouth and not in your hands. You know? Yeah.
And then the other method of increasing a capsule's shell's resistance to dispersal is to actually modify the material itself. So it's not a coating onto the capsule shell, it's a change in the material of the capsule shell itself.
So now the other question is what are, I mean, we know that, it's a probiotic. We know that it's something that is deemed to be beneficial for your gut, or some of the, you know, there's I think, how many do you have? How many strains? It's like
in the current version, it's 11.
11 strains in the current version. Okay, so yeah, you've modified it over time. So if you haven't used it in a few years, you might want to give it another shot.
Yeah, that's true.
But is it like dead, or what is it? What is it? A spore? Excuse me.
So the powder, which is made up of bacteria from two genera, Lactobacillus and Bifidobacteria, is lyophilized. Now, a more common term for this phrase is freeze-dried. Now, it's not really freezing, it's more drying, but it's called freeze-dried because it renders the bacteria in a state which is similar to desiccated food. Desiccated food is dried out, basically. Now, the purpose of drying out bacteria is to halt the metabolic activity in the cell, because without what's known as the universal solvent, aka water, chemical processes don't occur. And without chemical processes occurring, the bacteria won't expire. They won't like use up their internal reserves and then die. Now, it's very fortunate actually that this is even possible. It's not some foregone conclusion that you can take a thing, dehydrate it, and then it will reanimate when you rehydrate it. It's pretty fortunate for the whole industry that probiotics can even be made into a dry powder form.
There's in terms of humans, there's vampires. I think you can reanimate them, but it takes blood, right? Not
That's
water.
Right. Yeah, got to rehydrate them somehow.
Yeah.
The problem with freezing, just in case anyone is interested in why you can't freeze it, well, aside from it being a logistical obstacle in terms of maintaining the frozen temperature throughout the whole logistics chain, that would be obviously a big hassle. But the other reason is that when you freeze water, quite uniquely, it expands. Yeah. So it will actually split the cell.
Burst the cell membrane.
Exactly. Yeah, exactly. So that's why if you ever put an apple in a fridge, or certainly in a freezer, but if you put it in a fridge and it's towards the back and it's too cold and it actually starts to freeze, when it thaws out, the apple will be mushy. Yeah. And it's simply because the ice crystals have smashed up all the cells inside.
Yeah. Okay, so now, if you have, explain first what people get when they buy probiotics most of the time. I mean, we've talked about this before. I've certainly mentioned in my blogs that a lot of these people who sell probiotics are not probiotics manufacturers. They are they're packagers and designers of labels and marketers. Is that because
that's correct?
So they just go to there's any number of companies out there, and somebody walks over to the shelf and pulls out all these whatever strains they order and puts them into this thing in whatever proportions the specifications are called for, and then it mixes it up, pops it in a gel cap, pops it in a plastic bottle, puts a label on it.
Yeah.
And
exactly.
Boom.
So the majority of the industry is where you have these generic manufacturers and they do private label work because they just focus on the manufacturing. They're very big companies as well. So for example, in the USA, DuPont, you may have heard of DuPont.
Yeah.
They merged with Danisco, which is a probiotic manufacturing company, and they do some things in the US, and they do some things in the Netherlands. But they are approached by basically marketers who want to create their own supplement line, perhaps,
yes,
and sell it to an audience. So the only issue with this is that there's mixed incentives. So if you ever get in touch with these people, you will see that whatever magic ingredient list you've come up with, whatever magic strain list that you want, they will be very, very eager to actually influence your decision and say, okay, that's a nice list, but I tell you what you want to do is you want to go with this because we can get much higher yields with these strains, as in much higher CFU per milliliter of these strains when we're culturing them, and so on and so forth. And very quickly, a marketer or a supplement person setting up a supplement line will very quickly just be like, oh, okay, yeah, you guys are the experts, yeah, let's just go with that. And the manufacturers themselves have the incentive of just producing what they can produce in a good quality, but economically, and they can just get it out. And if it's selling them the same list of species which they're already selling to another 20 customers, then that suits them. So there's just a little bit of mixed incentives when you get an entrepreneur who doesn't really know what they're doing, using a manufacturer who has a lot of expertise and a lot of influence over them.
Okay, and so you have your own manufacturing facility and like I suppose some sort of warehouse like thing where you can manufacture it and hold inventory and so on.
That's right.
If
you think of Breaking Bad, have you seen Breaking Bad?
Breaking Bad, yeah.
You know, with the crystal. If you think of like those guys in the crystal meth lab, and that gives you a good idea of what it's like.
And so, you manufacture all 11 of your current strains here. Are some more difficult than others? Because it sounds to me like what you're saying is that the strains you use are determined by what you think is the best mix of products or mix of bacteria to seed into the gut with, not necessarily what's the easiest to make.
Correct, yeah. So the different species, the different strains have different potential yields that you can get in the manufacturing process. Now, the yield is just effectively the concentration that you can achieve of these strains in the culture medium prior to freeze drying them. And this is determined by their sensitivity to their own metabolic byproducts. So when you're culturing bacteria, if you want to achieve a decent concentration, then you actually need to buffer the medium, meaning you need to be modifying, tweaking the pH, of course, tweaking the temperature, of course. These are just basic things. But a good example is a pH. If you put in, bacteria can literally stop themselves from proliferating if their acidic metabolic byproduct isn't removed, isn't buffered. Now, some are more tolerant to this than others. So, for example, Lactobacillus bulgaricus, this is an example, is a very acid tolerant bacterial species. But there are other things which are not as tolerant, and so it affects the yield that you can get.
They're self-limiting in whatever medium they are as they proliferate and grow. Acid is one of their byproducts. So the medium gets to acid point where they maintain basically an equilibrium and don't just keep growing and growing and growing. Is that correct?
It's like if you were stuck in an airtight room. Eventually, your own CO2 is going to, poison yourself to death. You have to remove your waste products from the environment. And the other consideration during the entire manufacturing process, especially for bifidobacteria, that genus.
So this is sounding strangely, I don't think I ever thought about this before. Just as you're talking about it, I'm thinking, am I talking to a probiotic manufacturer or a cheese maker? Because it's almost right.
Yeah. You're right. You're absolutely right. I think a lot of the industrial know-how comes from the dairy industry. You're absolutely right.
So what medium do you start with? Is there different mediums for different ones, or is it some sort of medium that's all the same, or what?
Well, no, it's all the same. I mean, the core thing is all the same. So people ask me, people sometimes get in touch and say, is this grown on milk? Is this grown on some other thing, like because they're allergic to maybe milk protein or something? Now, despite the fact that Lactobacillus acidophilus is associated with milk and all these other things, you don't grow in a lab. You don't pour milk into a solution and grow bacteria that way. You use fairly standardized culture mediums, culture broths, which generally contain glucose, lactone, and a few other things, a few other sources of nitrogen, sources of carbon, and things like that. There is a little bit of proprietary aspects to this, but essentially you need a source of nitrogen, amino acids, a source of carbon for energy, and that's how you proliferate bacteria. They aren't choosy. Glucose causes almost every bacteria to proliferate, and glucose is a very well-characterized substance to use as the carbon source.
Cool. And so how do you, explain a little bit about your okay, so you manufacture these batches. I don't know if you do them one at a time or concurrently or what, but that's not really important. important point is once the process is finished, what's your quality control? How do you know that I've got the stuff and it's within whatever, parameters that I've established that's good quality?
So downstream of the freeze drying process is where you want to take the CFU count, because that's what the final product is in the form of a dry powder. So you can do various quality assurance processes at different stages, but what you want to do is you want to do it after you've converted it into a powder. And the acronym CFU stands for culture forming units. It doesn't stand for units. It doesn't stand for bacterial units. It stands for culture forming units. And a culture is multiple. Now, where this actually derives from is the fact that the way you actually quantify the bacteria is you take let's say you take a small amount of the powder and you heavily dilute it. Now, the reason you heavily dilute it is because you want to you dilute it serial dilution, meaning you dilute it multiple times. So by a factor of 10, by a factor of 10, and so on, until it's a thousandth or even more dilute of the original powder solution. And the reason for that is because you want to be able to take a small volume. Like when you
cut, when you have pure cocaine, but you don't want to sell pure cocaine. Wow.
That's exactly how I learned it. Yeah, that's where I learned it from, my experience selling cocaine. So you serially dilute this thing until it's magnitudes more dilute than the original solution that you put the powder in. And the reason you do that is because you actually want very, very, very few bacteria in that, very few remaining bacteria in that. And you take that solution, you spread it over a growth medium, and then you leave it, and you allow it to proliferate. And the reason you allow it to proliferate is so you can see it visually. I mean, you scan it with a computer, but in the olden days, you were doing it so you could see it visually. Now, you aren't seeing bacteria.
Like a petri dish culture.
Exactly. Yeah. Exactly.
You just do a little thing like that, and then it goes boom, and you can see what it is.
And if it's diluted, and you see these patches, right? You see these patches of cultures.
Yeah.
And if you've diluted it enough, then you can make a credible assumption that each one of those cultures was from a single bacteria, not from loads of them, because you've diluted it so much.
Okay.
And from that, you simply count the cultures. So let's say you had 5 cultures, and that was from 1 milliliter, but you had diluted it 100 times. This is just made up variables here.
Just math.
Then you can reverse calculate it and find out what was the original concentration, and does it match? Well, does it exceed what I'm aiming for with my batch?
Cool. All right. So, what's the business been like since you started? I'm not sure whether when I started putting on my blog and we started working together with it, I'm not sure whether you had just then started or very, very recently, but here you are 6 years down the road. What's it been like for you?
Well, I had recently started when I got in contact with you, and you were extremely helpful, to put it lightly. The first mention that you made of Elixa, I believe, was around April. I think it was like April 17th, 2015, somewhere around there.
Yeah.
I haven't looked this up. I can actually remember it. But you mentioned it so briefly just to begin with. It was just something along the lines of, you've tried Elixa, or you tried Elixa and you've had an epiphany, and then you talked about other things, because the article wasn't about Elixa, it was other things. And it was only one sentence on Elixa, and the amount of US interest that created, US sales overnight, was so surprising. And then from there, of course, you did some more dedicated promotions and really kicked off the US side of the market.
Yeah, I know. And at one point along the way, I don't know, maybe it was a year or 2 years later, or something like that. You were telling me that you get a substantial number of people. I'm not sure whether you're talking about all customers or customers that could come from my thing or what, but it's a pretty high number of people. Don't just get it once. right?
That's correct, yeah.
Right.
They maybe start with a single pack, because I sell it in single pack, double pack, 4-pack, and they'll maybe start with a single pack, which is something I encourage. I don't want to sell people something that's not going to help them. So I encourage them to start with a single pack. They start with a single pack. They see the results for themselves. They don't need to buy into any science or buy into any faith or hope or anything like that. They just need to see the results for themselves. Because I tell everyone that if it's going to work for you, you will see it within 6 days. On rare occasion, it might take 2 packs for you to see something.
And that's why, you sent me 2. It's been like a couple of weeks now that I got them. It took, from your shipping facility, which I guess is in UK, to here in Thailand.
UK, yeah.
In Thailand, 3 days. I was blown away.
Yeah, I was surprised.
I expected it to be a week, 2 weeks, something like that. But that's another thing, is that this is, a lot of manufacturers, maybe because I don't know. I won't speculate, but you get these bottles of probiotic and you take 1 or 2 capsules a day, and it's a 30-day supply. And then you want, basically you order 3 bottles at a time. But the point is that they're intended to be a constant supplement. And Karl never intended that. In fact, when I remember at first, it was 3 days. Three days,
yeah.
Right? It was 3 packs, right? And then you kicked it up to the 2. So this is 6 days. You take it for 6 days. And like I said, I had him send it to me because, so this is my own personal thing. It was, I had a bit of a head jolt, and something shook loose or something, but I ended up with what I thought was a sinus infection, but it wasn't one of those ones that just knocks you down. It was kind of just like persistent under the surface. And after a while, I thought, well, maybe I'll take a round of antibiotics. And I did, and it fixed it. But in terms of the gut, it just kind of made things just not great. And I figured, that'll fix itself. But it didn't. It kept going on, especially if I had some booze or something, then it would be kind of awful. But so I get Karl's thing, and 1 dose, one out of the 6, daily 6. So this is really kind of a dose, I guess you would say, but 1 treatment. Yeah. First treatment,
one sachet.
1 sachet, which is 10 capsules. You down them, and next day I was better. Well, of course, I finished the dose, but I'm keeping this one. What's the shelf life? I won't keep it around forever, but I think, what would you say, Karl?
It's at least 2. It's at least 2 years at room temperature.
Yeah. So if you take a dose, a 6-day course, once every, I'd say once every 2-3 months, you're probably going to be fine, unless you have a specific problem, like after a round of antibiotics, or after a serious weekend of binging that lingers, booze binging that lingers. I don't know, how badly does alcohol consumption affect the gut? Do you know?
Well, yeah. So ethanol directly impacts the permeability of the intestine, and so it acts in a very acute manner, in that you can ingest ethanol, and within a short period of time, the concentrations of ethanol in the lining of the intestine match the concentrations in the bloodstream. And the best way of describing it is an experiment that was done on mice. Now, I've always thought that liver damage caused by ethanol is not the direct effect of ethanol on hepatocytes. Ethanol is directly toxic on hepatocytes, on liver cells. But some people, as you know, they can drink loads and they feel fine. Maybe they don't even get a hangover. Other people, they drink a little bit and they'll get terrible hangovers. Yeah. So there's obviously some reason for this. Now, the reason is because ethanol increases the permeability of your intestine. So if you're already dealing with a dysbiosis, it's simply going to increase the translocation of inflammatory material going from that dysbiosis into your bloodstream. First port of call, of course, is the liver. So when blood absorbs things from the intestinal tract and moves throughout the body, the first port of call is to the liver. So the liver can detoxify what has been absorbed and a few other things. So what this means is that the liver gets the brunt, the tidal wave of translocating inflammatory material that occurs when you drink alcohol. And they proved this quite definitively using mice. They gave mice ethanol and it caused liver damage. Then they gave mice a large number of antibiotics to completely kill all the bacteria in the gut. And they gave them ethanol at the same time. And it didn't cause the liver damage because there was no dysbiotic bacteria to translocate material into the liver. Then, as a final check, they infused the blood directly with something called lipopolysaccharide, which is endotoxin from bacteria. So there was no alcohol, there was no antibiotic. It was just a direct infusion of LPS. And this caused the same liver damage that the ethanol alone caused. So those three experiments proved that it wasn't the ethanol, it was the ethanol increasing the movement of inflammatory material from the bowel into the liver. That's what causes liver damage with alcohol intake.
Wow. It's interesting. So, okay. Now let's talk about food. So obviously, if you eat, what we call, a crap diet, you might have more need of these than if you have a healthy diet. I remember one of the early on posts we did was about your times in Uganda. And you had pictures of some of the meals there. You want to speak to that a bit?
Yeah. So in rural Uganda, where I spend most of my time, the kind of food that they eat is very unprocessed. So it's not been processed via factory machines. If you see the food, you will recognize where it originated from. It's not in a box. It came from a tree or it came from the ground.
That's a potato, that's a carrot, that's an onion, exactly. That's some pork, or that's some beef.
Exactly.
Right. That's some broth, that's chicken, right.
Exactly.
Go figure.
And they tend to focus mostly on sources of starches, rather than fruit. There is fruit abundant in Uganda, but for a growing population, like entire civilizations have grown on starch. That's an undeniable thing, whether it's healthy or not. Yeah. Entire civilizations have grown on it because the density of humans is so great. Like we are such a prolific species that we need something like agriculture to fuel us. So a lot of what they eat is cassava, maize or corn, which is sugarcane. They don't eat too much sugarcane. Potato, which they call Irish potatoes because they're referring to the white potato. They also have sweet potato. But their the iconic thing in Uganda is matoke, which is green banana. Now it's not unripened banana. It's green. It's just green banana, and it's not plantain either. And it's quite hard. So what you do is you peel it and you steam it, and you mash it up. So it eventually becomes a bit like mashed potato, but it's sweeter, and it's very tasty, and it's very healthy as well for the gut, if you've got a healthy gut to begin with.
Yeah, that's an interesting thing. So if you have a healthy gut, meaning it has sufficient populations of the widely recognized beneficial bacteria, then of course eating unprocessed food with various degrees of fiber from starches and grains and so on I don't think anybody disputes that would be harmful. But how about if you have a really crap gut and you eat a lot of food that is, I guess the term is fermentable or what?
Yeah.
Okay.
That's right. So basically, two ways of looking at it are like this. One way is if you have a gut dysbiosis, everything that you can conclude from an evolutionary standpoint goes out of the window. So you can be very correct about what is an evolutionary intended diet or an evolutionary natural diet for a human to eat. But if you have a gut dysbiosis, it goes out of the window in the same way that if you have a torn bicep, everything you know about how biceps strengthen from applying weight in the gym and so on, everything, all of that goes out of the window because you're dealing with such a dysfunctional system to begin with. The other way of looking at it is a more simplistic metaphor, is that in fact, it's not even metaphorical, it's literal. If your gut is filled with bad bacteria or bad microbes in general, then eating highly fermentable fibers, such as the residue of starch, even sugar, because these are present in the small intestine, and non-starch polysaccharides, this will simply feed them up. If you have a gut full of good bacteria, you can fuel them and feed them up with fiber. If you have a gut filled with bad bacteria, you will similarly fuel them, feed them up, cause them to proliferate from eating fiber. And this is why people with a gut dysbiosis can find that when they, the more they focus on their health, the more they eat their greens and all these other things, the worse they can actually feel.
Yeah, I've had tons of people, and then there's what is it, FODMAP diet and things like that. And so yeah, that's heard that a lot, and it makes sense if you think about it. If it's, and that the bicep analogy is good. So if you got to fix the problem first. So it sounds to me like, and then I'll have a follow-on question here because it just occurred to me. So it seems to me like if you have the bad gut, there's not much way to rid yourself of the bad bacteria. What you do is you take the probiotic and get the good bacteria up. Then what happens? Then what happens to the bad bacteria then? So say you're successful in taking a probiotic product. Say you limit your fibers and stuff for now, so you're not making it worse. And then you take sufficient numbers for a sufficiently long time to get the good bacteria up, levels up. Am I on the right track here? And then what happens? That's right. Then what happens?
So essentially, using a probiotic, the way I view it, a lot of people try to claim that it's replacing missing species into the gut, which would imply that the way it works is that you have a certain species missing from an ecosystem, and you return those species in the form of a probiotic pill. And by returning them to the species, the rightful occupants of those ecological niches, they will competitively exclude the bad bacteria. However, I think based on my experience,
keep going.
Sorry, based on my experience with Elixa and the results that people have had, and how important it is to have a high dose compared to just a small dose of the same species, I think it's apparent that the way that a probiotic works, such as Elixa, is as a targeted antimicrobial. And all that means is it kills other microbes, but it does it in a targeted way, unlike an antibiotic.
So it's like a war zone down there. So taking probiotics, specific ones that are like, what are they, like really good, infantrymen, or are they bombers with nukes, or what are they,
Well, you actually did an article with me, and you came up with the title, as I remember, FODMAP Mercenary, FODMAP something and Mercenaries, right?
Mercenary, yeah.
Yeah. And so that's effectively what it's like. These bacteria produce bacteriocins and fungicins, which simply means chemicals which kill other bacteria and fungi. And these can be quite simplistic. They can be hydrogen peroxide,
but not themselves. But not themselves, right? So they're
Exactly.
They're immune to their own. It's like a snake bites itself, no problem, right?
Exactly. Not only that, but these substances are deployed in a manner which doesn't target beneficial species in the gut. Because if they did, it's not simply something I'm asserting. If they did, then these species would not be members of a healthy ecosystem in the first place.
Right.
If it's by definition, if it's a member of a healthy ecosystem, then it can't be something that's going and destroying every other species in the ecosystem. So they produce acids, they produce hydrogen peroxide, and then they produce more complex bacteriocins. They can even infiltrate into biofilm communities. There's quite a few more complex things that they do, but they effectively use chemical warfare.
That's great, yeah. And so now, but it's probably, I would imagine that it's not like they're trying to sterilize the gut or anything. It's what they do, their activity keeps the bad bacteria down to levels that don't bother you. I've heard even that if they test vigorously enough, they can even find C. difficile in certain people, but it's just that it's always kept down at a low level.
Exactly. When you have a slight change in an ecosystem, for example, if we think on the macro scale, like wolves and other animals, if something happens where the population of one species just decreases a lot dramatically for a short period of time, or maybe increases a lot, then it can set in motion a chain of events where all the other ratios of species become deranged. Now, so with the example you gave is Clostridioides difficile. Now, it used to be called Clostridium difficile, but it's recently been called Clostridioides difficile, in case anyone's wondering why I'm calling it a different name. But this can be present in a healthy human gut, but it's kept repressed, it's kept suppressed because it has its place in the ecosystem. It has its own little niche and it stays in its lane. And it's suppressed by the fact that there are all these other healthy species which are occupying the other niches in the ecosystem in the gut. Now, the problem occurs when you take antibiotics and wipe them all out. Now, Clostridioides difficile, which can be resistant to these antibiotics, has an open field. It has a blank slate to now proliferate into, and to occupy. And you might think, well, when you stop the antibiotics, why don't the original species just regrow? Well, there's two reasons. Firstly, they may have been completely extinct, and in which case they obviously could not regrow. Secondly, is what allows a problem to occur, it doesn't necessarily fix a problem when you remove it. So I use the analogy of smoking. If you smoke all your life and you develop lung cancer, you can't simply stop smoking and then the lung cancer will resolve itself. Yeah. So once the Clostridioides difficile have expanded and occupied all these different niches and in fact modified the expression of genes and a lot of other things, it's much harder now for the original species to supplant them from the gut. So yeah.
That's okay. Good. Now, let's see where, I don't know how much I think in email yesterday, you said you're working on another project, and do you care to share anything about that at all? Or we can say, no, it's top secret. It's fine. It's up to you.
No, I can certainly share some things. Firstly, what I want to say is that you've known me for a while, and you probably know by now that I'm not so much a marketing promo guy, and I'm a lot more interested in developing.
That's why I like
the product.
That's why I like
and the development of Elixa to date has been really just the tip of the iceberg. Changing strains, changing species, modifying the production technology is useful, and it's created improvements. But it's really small compared to what I'm planning next, which is not just a blend update or something like that, but is a whole new product, basically. So it's still a probiotic, and it still works exactly on the same principle of correcting gut dysbiosis. And gut dysbiosis is still absolutely the number one thing that needs to be corrected to solve basically 90% of chronic ailments that people are suffering from. But as some of your audience possibly know, and as I think you're aware of, there is growing literature surrounding the use of transplants from a healthy person's gut into an unhealthy person's gut. So in other words,
you know, I was getting that close to making a joke about what you're doing next, about fecal transplants, but I thought, well, I don't know. And now here, so yeah, I seriously had no idea. Okay, go ahead.
So the holy grail of these this field would be to create it in pill form, of course, to make it something similar to Elixa, where it can just be bought online, shipped, and taken. But there are a lot more obstacles than simply being able to freeze dry the donor material from a healthy donor. And this is evidenced by the fact that there have been thousands, if not tens of thousands, of FMTs performed, which, just to elaborate on, is where you take a sample of the microbiota from a healthy individual by taking a stool sample, and you actually inoculate an unhealthy person's gut with this bacteria. Well, actually not just bacteria, but many microbes. Now, this has been done so many times with so many variations from so many different donors that it's quite clear that, firstly, it can be extremely effective and work miracles. Secondly, it's not at all reliable or a guaranteed success based on all the variables that we don't know that affects the effectiveness of the procedure. In other words, the engraftment of the donated microbiota into the recipient. So there are a lot of ways you can tweak the protocols. Sometimes people have tried taking, there have been clinical trials where you take it orally in capsules, where you take it via nasogastric tube, where you take it via enema, where you take it by a colonoscopy, which is a bit like an enema, but it's put all the way into the cecum. So there are various ways that they've administered this microbial consortium. And they've even modified the preparation protocol. They've done things where, like, they've given people lots of antibiotics to clean the slate, so to speak, or fasted, and so on and so forth. But
enemas and things like that, right?
Exactly. Yeah. So the bottom line is this: this is an extremely promising treatment modality, and I am absolutely convinced that the concept is the solution. The only thing is, it's not been no one's gotten it to work properly yet. And what I mean by properly is reproducibly across all of the conditions that gut dysbiosis causes, and permanently. That's what I'm working on.
I have a friend who's well known in the paleo community. She's public about it, so it's no Karen, and she claims to this day that she did a DIY transplant with a donor and changed her life, like, overnight.
Yeah. Absolutely. Yeah, I mean, the potential, this is where it's at. Now, people shouldn't be thinking, okay, so FMT is where it's at. What I'm saying is, inoculating an unhealthy recipient with a full spectrum consortium of human intestinal microbes currently being sourced from another healthy human, that's what the fundamental core concept is. That's going to be the future of this medical field. Now, whether that comes from an actual human, whether it's cultured in a lab, whether it's in a sort of raw form, whether it's in a refined form, meaning it's been centrifuged and separated out and things like that, that's all. There are many people in this field. It's kind of like a technology race. You
That's
know,
where
the beginning of social.
that's where the science, engineering, and technology come into it, which is where you come into it.
Exactly. Yeah.
Okay. That's great. Okay, we're quickly coming up on our hour here, and I think hour is a good amount of time for a video like this. Anything else to add to this, Karl?
Probably nothing quick. And anything else I would probably be another in-depth discussion. But I don't think I do have anything. I don't think I have anything to add. Not off the top of my head.
Then let me wrap it up here. Karl and I have been talking. We're probably going to put together a bit more of an elaborate page on my website, and it's probably going to rely heavily on real testimonials from real people. Karl mentioned he's not big into the marketing and promotion stuff. And I said, that's why I like him. I've always liked this because it's just like, hey, here's the product, try it. You like it, order it again, try it again. You tell somebody about it if you think it might help them. But this endless hype, which always seems to relate that, there's got to be something perpetually wrong with you. You've got this problem. I've got a pill for that. I've got this for that. I've got this for that. And, I just so hate it. I hate that you have to not only get up and lie, but you have to lie every day. You have to keep lying. And then you have to have bigger and better lies because the old lies get tired. Can't you just say, hey, I think it's a good product. We do change it out sometimes. We started with this many strains and now we're up to 11, and we take feedback. If someone who has a bad reaction to it, try to figure out, well, is that just like a crazy one-off thing, or is it something that I can learn from? So on and so forth. That's how you do solid business with a long-term view. Right? Not like, I'm going to get in there and make my millions now before blah, 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 blah. You don't have to worry about the competition. You have to worry about having a good and better product. Anything to add to that, Karl?
I would say that I certainly agree that that's the approach I've taken. I think you do that on your blog as well. You encourage all kinds of feedback. It's not just, I only want to hear good things, or I only want to hear people who are towing the party line. You encourage people to discuss bad reactions to maybe resistant starch or anything, and just discuss it. Don't take it personally.
There were some, but the thing is what that does is it tends to give people more confidence that the ones who say, yeah, it's been great for me, it's like genuine. If all you hear is, it's great, then it just, hell, there's tons of pharmaceutical drugs out there that are lifesavers for some people, but that doesn't mean that some people don't have a negative side effect to them.
Yeah. And what you said about you're sick of people like saying you've got this problem, you've got that problem, you need this pill, you need this pill. I agree. People get fatigued. Especially people who aren't maybe as informed as you and I, they'll be researching online and they'll just get frustrated and fatigued because there'll be so many. As soon as they've learned about one thing, there's this other thing that's a problem. We need to keep it simple and say this: humans haven't been, walking around for hundreds of thousands of years having to take pills. Once you can correct the system back to the healthy equilibrium, there's no evolutionary justification for having to continually take something.
And the thing is all the search engines are so corrupted now, because it would be like, literally, if you want to find out about something, say you go down the street and there's this place and you go in there and they say, I'm looking for information about this and this and this. And they say, well, let me show you here. These are all the people who pay us to give you their information. This one pays us the most. So we want you to look at this one first. right? And this and this and
Yes.
this. So
Exactly.
and it's fine. That's a business, right? That's a business, but people go into this thinking that like Googling something or Binging it or whatever, you know, even DuckDuckGoing it is going to somehow deliver them, you know, a fair and balanced thing of results. Meh, don't think so. But you can certainly get some information if you try. So.
Agreed. Yeah.
All right. So with that, and Karl, stay on with me, but I will get this crunched and up on YouTube fairly soon. Thanks for watching. Thanks for listening. Again, freetheanimal.com. You'll find Elixa right on the sidebar. So you just click on it. Or if you want to go there direct, just go to elixafta.com. FTA is in Free the Animal. So Elixa FTA, ELIXAFTA.com. There you go. All right. Thank you. Bye.
Thank you.